Hi guys, today I'll be talking about lipid absorption and lipid digestion. But before going through this, we need to know what kind of fat exists in our food. Okay, highest amount of fat in our food are called triacylglyceride, and small amount of fat are from cholesterol and phospholipids. Now, we have three areas in our body that are responsible in digestion of lipids. We have mouth, we have stomach, and we have intestine. Now, in the mouth, at the dorsal root of the tongue, we have glands that secrete an important enzyme called lipase. This lipase only works on a short triacylglyceride. What does short triacylglycerides means? We have two types of triacylglyceride. We have long, we have short. Short means the triacylglyceride having a chain of carbons which are less than 12. Long, that means triacylglyceride will be having a chain of carbons which are more than 12. Now, so lipase will be working only in triacylglycerides which are short, that having carbon chain that is less than 12. Now, after finishing from mouth, we will be having products such as fatty acids from the short triacylglycerides, and we will be having long triacylglycerides and cholesterol and phospholipids. Now, going to our esophagus reaching the stomach, our stomach cells will secrete also the same enzyme that is secreted in our mouth and is called also lipase, and it also works on short triacylglycerides. So when we are finished digestion lipids in our stomach, we will be ending by long triacylglycerol, cholesterol, and phospholipid plus free fatty acids that are derived from the digestion of the small triacylglyceride. Now, finishing from stomach, going to the intestine. When we are going to these contents enter the intestine, as we know, they'll be carrying an acidic pH because they were in the stomach, and in the stomach there is hydrochloric acid. So when they go to the intestine, they'll be carrying this environment. This acid will affect the intestinal cells, which are called enterocyte, and when they are affected by this acid, they will secrete a feedback hormone called secretin. The secretin will stimulate the pancreas and will stimulate the bile to secrete something called bicarbonate. This bicarbonate will neutralize the acidic pH, because acidic pH is really dangerous because it might break down the cell junctions, and breaking down the junctions would make a lot of bad complications. So after neutralizing the pH, we'll be having in our intestine cholesterol, phospholipid, long triacylglycerides, and free fatty acids derived from previous digested small triacylglycerides. These small fatty free acids that are derived from short triacylglycerides will be absorbed directly by the intestinal cells. Why? Because they are small enough to be absorbed. When they are absorbed, a feedback hormone will be secreted by the intestinal cells called cytokinin. Cytokinin will stimulate the bile to secrete its own salts, and the function of that to emulsify the fats, because fats need to be emulsified. Why? Because they are nonpolar, and for the fat to be absorbed, there is a required condition, which is the fat should be in a polar structure. So the bile salt will come from the bile and will act on the fat by increasing their surface area, so that they will increase the affinity of the enzymes that are about to work on these lipids. Now, we said cystokinin will stimulate the bile to secrete bile salts, and cystokinin will stimulate the pancreas to secrete the hydrolytic enzymes that are required to digest the fats, long fats like long triacylglyceride, cholesterol, and phospholipids. Now, let's talk about each fat digested, like triacylglyceride, how it's digested, phospholipid, and cholesterol. Triacylglyceride is digested by two types of enzyme. One that, by the way, they are shortly secreted by the pancreas. Okay, these two enzymes are called esterase and colipase. What does they do? They take the triacylglyceride, and as we know, triacylglyceride is made out of 3 amino acids plus glycerol. These two hormones will break down the first fatty acid and the third fatty acid So we will have a product of 2 free fatty acids plus 1 fatty acid attached to the glycerol. So this fatty acid that is attached to the glycerol is called monoglycerol. It will be called monoacylglyceride. Okay, monoacylglyceride. So the product of digestion of triacylglyceride is 2 free fatty acids plus monoacylglyceride. Why it's called monoacylglycerides? Monoacyl means 1 fatty acid, glyceride, the glycerol structure. So we'll be having 2 free fatty acids and 1 monoacylglyceride. Now let's go to the second step, which is digestion of phospholipids. Okay, pancreas will secrete an enzyme called phospholipase. Phospholipase will act on phospholipid in this way. See, now phospholipid, as we know, it will have 2 amino acids and 1 phosphor group, and these 3 will be attached to glycerol structure. Now, what does phospholipase do is breaking the bond of the first fatty acid. So we will be having a glycerol structure with a 1 fatty acid and 1 phosphor group. So we need to break the other fatty acid. What we will do is having another enzyme called lysophospholipase. This lysophospholipase will act on the second amino acid that is still attached to the phosphoglycerol structure, and we will break it. So we'll be having now 2 free amino acids and glycerophosphate structure. The glycerophosphate structure may be degraded, may be absorbed, maybe will be excreted into the feces. Now let's talk about cholesterol. Cholesterol exists in 2 phases. We might have it as a free cholesterol. We might have it as an esterified cholesterol. Now, there is an enzyme or pancreas will be secreted called cholesterol esterase that will only work on esterified cholesterol and break it down. And what does esterified cholesterol means? Esterified, we know, it's a process between alcohol and fatty acids to produce a fats, right? So esterified cholesterol, we need a cholesterol esterase that break down the esterifying bond. So we'll be ending by free cholesterol and fatty acids. Then the free cholesterol will be easily absorbed, and the fatty acids, because they are small, will be easily absorbed. Now, when we finish digestion of these 3 fats, they will not be easily absorbed directly by the intestinal cells. What will happen is the bile salts that exist in our intestinal lumen, the products of digestion of lipids that also exist in our intestinal lumen, plus the fat soluble vitamins, which is vitamin D, vitamin E, vitamin A, vitamin K, that also exist in our intestinal lumen, will all combine together and form a ball shape. The exposed surface of this ball is polar, and the internal structure of it is nonpolar. And as we said previously, in order these compounds to be absorbed, they need to be polar structure, almost polar structure. And because their exposed surface now is polar, the intestine will recognize it as a polar structure and will absorb it. This ball structure called mycel. This mycel contains soluble fatty vitamins, as we said, the product of digestion of fats, and the bile salt. They will be absorbed by the intestinal cells. The intestinal cells will use it, and we will resynthesize the triacylglyceride and other stuff. Thank you for your watching, and I hope my video helped. Bye bye.